The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 8th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the weekly bioanalysis, which features news and podcasts more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotti, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian, and I've been in the flow cytometry space for over 13 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with an additional location in Europe, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri, so we're truly all over the map. We are elated to have you listening to the 8th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will feature a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback received from you. We're constantly looking for topics and would be happy to discuss something that you want us to cover, so please reach out. So again, we're fired up to have you here and are looking forward to a fun episode today. To kick us off for episode number 8, Brian is going to go over the news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got a full docket of medical news, investments, and KCAS news today. So in terms of medical news, the theme of today is beyond, right? And we've been talking a lot about immunotherapy and CAR T on and off for the last couple episodes. So today, we're talking about beyond TYK2. So TYK2 is one of those immunotherapy markers that a lot of different companies are currently targeting, and there are a lot of different trials ongoing to understand how to modulate that activity. So beyond TYK2, there's a story out talking about the upcoming ASCO conference, which is going on this weekend in real time. And it's talking about what it thinks is the next TYK2, which apparently Nimbus Therapeutics thinks is HPK1.
Mm-hmm. Right. And, as far as TYK2 is concerned, the importance of it, looking at cytokine signaling, right? It's part of a pathway, an intracellular pathway that when activated can produce proteins that then guide cells to do certain things, right? Whether they're activated, they proliferate, so on and so forth. And so with HPK1, it's hematopoietic progenitor kinase 1. It's for immune cell expression, T cell signaling, right? And so by blocking it allows for tumor suppression.
Exactly. And, these immunotherapies are incredibly powerful tools where we can sort of fine tune exactly how the immune system works. And this is sort of another method. So you mentioned cytokines. So that's, one of the primary ways that immune cells can release soluble factors to impact, tumor growth and different immune system functions. And here it's actually regulating the activation of the cells themselves as opposed to the cytokines.
Right. And interestingly, HPK1 could also be used as a predictor as far as treatment with immune checkpoint inhibitors. And so whether the presence or absence there can guide whether or not the treatment would be likely to fail or succeed.
Interesting. I had not read that far down in the paper yet, but yeah, that's we'll definitely have to look more into that, being a bioanalytical group, right?
Right.
So I found this to be very interesting, and hopefully another round of companies can grow up around sort of the next group of immunotherapy targets, because definitely we've seen incredible results from the first round of immunotherapy targets, first in oncology and now in autoimmune. So hopefully with these new proteins, they'll give another sort of druggable target there.
Absolutely. I have more success with those checkpoint inhibitors to be able to attack those cancer cells.
Yeah. And while immunotherapy is, I would call it a relatively mature field, because the therapeutics that we're using to fine tune the immune system are either small molecules or some biologics that are, we know how to characterize, we know how they work. Definitely, the CAR T space is still a little bit more the Wild West, where it's a much newer technology. And even here, we're seeing groups that are saying, we've done BCMA to death. Now we want to go after other types of receptors that we can modify.
Right. And the importance here is you're always looking for what's called a tumor specific antigen. The reason being is that gives you more homing ability and less off target effects. Whereas a lot of the proteins are what's called tumor associated antigens. Now, these may be more frequently expressed on cancer cells. I should say they are more frequently expressed on cancer cells. However, there is some expression on other cell types. And so what you have to do is investigate which, if hitting those other cell types happens, what is the effect, right? And so that's where you always have to screen out for the ideal candidates there to attack just what you want, have the effect that you want, and limit any side effects.
Yeah, that's exactly the right way to think about it. Basically, the more tools you have in the toolbox, just like with immunotherapy, same thing with cell therapy. The more different types of cell therapy you can have, the more different types of cancers you can treat. Every cancer, to some extent, being relatively unique. So, this story and sort of discussion point that we listed here for the medical research news is targeting GPRC5D as one of the most promising next steps beyond BCMA there.
Yeah, it's interesting reading a little bit about it being an orphan receptor. And so it's similar to other receptors, but hasn't had a ligand identified. So the protein that would bind to it. And so it makes it a very interesting target there. One of the other things I wanted to point out too, we're talking about therapies, and we've gone through before bispecifics and chimeric antigen receptors, and just one of the things as I was reading up on this specifically was how do bispecifics and CAR Ts, how are they differentiated, right? Obviously, we've got the cell arm of CAR Ts. We are putting a targeting feature on the surface of the cell with some sort of activating domain intracellularly. However, with a mAb, bispecific specifically, you will try to recruit the T cell to bring it up to the tumor cell for some sort of killing activity. When you have a CAR, you're still targeting that tumor cell, so you still have that tumor targeting arm. However, you're bringing the whole T cell with you. So, essentially it's like, here it is, now go bash it, right? And so I just like to think about it in that way.
Yeah. Yeah, no need for that homing mechanism, right? So I guess it makes it a little bit simpler. Now, you are attaching, of course, the whole cell to it, but, at least you don't have to worry about that recruitment part.
Exactly.
Nice. All right, definitely, but very exciting news both for immunotherapy and for cell therapy as we start to branch out into more different targets and more different classes of targets. And that leads directly into investment news. So here, the story that we've got for today is sort of the continuing pivot from oncology into autoimmune diseases. So a company called Grey Wolf is yet another one. I think we had two stories last podcast about companies that were pivoting from oncology to autoimmune. So now this Grey Wolf company is doing the same thing and has raised an additional $50 million of funds to be able to do that more effectively there.
Yeah, and they look at inhibiting endoplasmic reticulum aminopeptidases. And when I read about that, I was really interested in understanding. So, when you have a difference in your immune system between your T cells and your B cells, specifically your B cells have essentially antibodies on the surface that can recognize the full protein by binding to an epitope, whereas T cells don't do that. The proteins are processed within the cell, chewed up, and then they're presented on the surface as antigenic fragments, essentially. What Gray Wolf is looking to do then is modulate what kind of fragments get on the surface, and so they can then treat immune dysfunction both in oncology and autoimmunity in that way.
That's exactly right. And it's always interesting to see with just company development, right? That in some cases, you want the company to remain as focused as possible on a single indication to make sure that it can conserve funds to get all the way through clinical trials. But then in other cases like this one, it really makes a lot of sense to raise more funds and go after a wider scope of targets to sort of reduce the amount of risk for any one target, right? So maybe it'll work great for autoimmune diseases, but maybe it won't work quite as well on oncology. And then this sort of de-risks the company to allow it to, when it pursues more options here.
Right, right or wrong, I'm used to more when you're expanding the toolbox. It's when a company is getting more mature, they're identifying more uses for it, expanding. Whereas usually a younger company would be expanding its toolbox, right? And essentially making sure that the treatment is efficacious in a certain platform. In some ways, the company is then looking to get bought or something like that, looking to raise more money, and then going to that next step of expanding the various therapeutics. Because, like you said, I mean, the biggest point here is clinical trials are really expensive. And so when you're trying to look at other disease states, for example, it's going to be very costly. So you've got to raise those funds or partner with other companies to be able to do so.
Right. And towards that point, in terms of partnering with other companies, the other story we've got today is just sort of the ongoing string of announcements that we've had for most of May on large pharma basically reducing headcount. So I think it started off May with BMS, included Pfizer, and most recently, to this podcast at least, was some announcements from Takeda. But we've linked to a story that just tracks quite a few, specifically in the Boston and San Francisco area, that have been quite impactful. And it is, as you just mentioned, Adam, related to that pipeline reshuffling.
Mm-hmm. Yeah, and in my notes in prep of this podcast, I just have layoffs. I just have one word. It stinks. It really brings forward that human aspect from another side, right? All of us as scientists, we were in there to try to help people's lives, improve people's lives by getting them therapies to make them better. And those scientists are impacted, and not only the scientists, business teams, so on and so forth, by these layoffs, right? And it's something that certainly is a struggle. But I will say that one of the things that I've seen a lot more of, and I'm happy about, is that people share about jobs on various sites, whether it's LinkedIn or other sites, share posts that they're looking, and it's really a very positive atmosphere in that respect, that everybody wants to reach out and help one another. And so if there's any bright side to something like layoffs, it's just a recognition that, you don't only help as a scientist for creating a therapy, you help too from the personal level of just sharing somebody's looking for a job, or that there is a job posting, and making sure that those connections are made.
I would agree wholeheartedly. And, again, because these things are happening on such a macro scale, it really has made this type of layoff entirely lose its stigma, because it's nothing related to a person's performance that is causing this. It's because a company decided to drop an entire asset, and then they're making the changes because of that. So definitely, I've seen a lot more people, as you say, openly sharing this and then getting new jobs, and in a lot of cases, then starting new companies, right, and or
Hmm.
potentially taking companies in new directions. So
Yeah.
always an optimistic thing from that side too.
Curious as an aside, I know I was impacted by a layoff early in my career. So this was back 2004. No LinkedIn or anything like that. Doing the traditional job search, sending out resumes, things of that nature, going to job fairs. Curious, had you gone through a layoff before, Brian?
No, not entirely. We went through one merger where it got close, but always a concern, I guess.
Yeah, those things are stressful too. I had been through other rounds where the company was considering it, and you realize you got to keep your focus on the day to day, but at the same time, you're going home, you're doing your job search, and just covering yourself just in case. And so, certainly understand what people are going through in that respect, and wish for speedy job searches there.
Yeah, and hopefully we've reached out to a couple people, and hopefully we can pick up some new team members from that perspective too. So
Absolutely.
always a silver lining.
Hmm.
Um, Switching gears entirely, I did want to follow up on our last podcast, KCAS News, which was upcoming conference season,
Hmm.
and ideally declare conference season over. I know that's not entirely the case, but we did definitely attend Cyto with quite a few folks. We went to WRIB, we went to APS NBC. I went to Cyto personally, and of course, the topic of this podcast is flow cytometry. So happy to share a couple things there. And I know we do still have a couple more meetings. So we've essentially set up our own mini conference with SciTech here at the KCAS Bio Philadelphia site. So we're looking forward to that in June.
Hmm.
And then also we've got the AAPS Summer Scientific Sessions, which is happening right next to our KCAS Bio Kansas City site.
And now you said you went to Cyto, but you went to AAPS as well.
I went to APS NBC, and
NBC,
then there
right,
was
right.
also the WRIB that was going on like at the same time as Cyto. So a lot of people had to split their time and attention there.
Right, right. Yeah, you've done a lot of traveling there. And so tell me the thoughts on Cyto. I know you and I have been talking a little bit about it, but having attended it last year myself, really enjoying it, getting a lot of information out. Able to share like a favorite workshop or something?
Um, yeah, I think that the name of the game at Cyto this year was really around high dimensional analysis.
Hmm.
And there were quite a few workshops and series around how to include AI, how to deal with high parameter clustering, and how to really interpret data afterwards. So I think I went to probably 4 different workshops sort of around that workflow, and definitely an interesting problem of, now that we can collect 40 colors at a time from every sample that comes in, how do we actually make sense of that data after we get it?
Hmm. Right. I do remember there was discussion about that last year. I think that with the advent of spectral, realized that is certainly going to be an issue that we're going to need to address. The more markers you get in there, the bigger the data sets, and so finding those tools, and with the advent of AI and incorporating that in different formats, it seems like there's a lot of options out there right now too.
There are, and some of them are extremely accessible. So plugins to FlowJo, plugins to Omic, that are extremely easy to just adapt in our current workflow. And some of them, entire companies have spun up around proprietary algorithms. So we'll have to see exactly how the community decides to organize it. Definitely as an outcome, we've gotten involved in a couple of standardization efforts to basically bring some sort of order and progress to the field overall, to say, this is data that is ready for AI analysis, and this is data that is not ready. And I think that'll be sort of the most important first step in making sure that this AI is sort of seen as a modular additional service, as opposed to sort of a bespoke thing that needs to be planned for at the beginning.
And each one has its own unique features. I'd be really curious to see, because with data analysis, it's not standardized, right? From one study to another, one client to another, one project to another, you could see different ways to analyze data. And so AI is tasked with that, right? And the humans that are using AI are responsible then for teaching it how to go about analyzing. And so it makes for a lot of learning that's going to go on, as in the nature of it. But then also, how does that get simplified? Is there a way? So excited to see as that unfolds.
Absolutely. I guess we'll have to wait for next Cyto. But we'll also be at AAPS, that has quite a vested interest in understanding AI, and also this year probably ICCS, so that it'll be more of a clinically focused community as opposed to a cytometry focused community.
Right, that's the one that's beginning of October. Certainly looking forward to going over there. Seattle.
Yeah, indeed. All right. And with that, I guess we can sort of wade back into our paper that we've been going over for a while. And now we're sort of moving on to the last phase of it in terms of how receptor occupancy testing works in clinical trials.
Right. And so the paper that we're looking at, the reminder, is Cytometry Part A, multi-parameter flow cytometry assay for quantification of immune cell subsets, PD-1 expression levels, and PD-1 receptor occupancy by nivolumab and pembrolizumab. And so this works with a 12-color assay here, identifying key subsets and then looking at the expression level of PD-1 on the various subsets that they've identified. It goes along a lot with what we've been talking about, where it's important for receptor occupancy to identify the cell types that are important to you, but also to simplify the assay. Receptor occupancy can be somewhat challenging as an assay, and so you want to fine tune it as much as possible. It's not to say that you can't analyze other things. Certainly you can do a multi-tube assay. You can expand it and do more colors, but by and large, the really tight assays are of those 12 color designs. And so what is something that really struck you as far as the paper is concerned, Brian?
Really, in terms of the paper in general, was struck by at some points how variable the data was, but then also how sort of interpretable the trends were at some point as well. So in some cases, the values were going with CVs as high as 100%, and then in other cases there were some extremely tight CVs of around like 5 to 10%, depending on the size of the population and then the frequency of the expression of different antigens there.
Yes, and I think one of the keys is something that we've really latched onto, as well as utilizing beads to analyze PD-1 numbers, to aid it tracking from sample to sample, batch to batch, and then also following up the validation with testing on disease state samples to confirm that things are looking as they should. And finally, like the specificity testing to ensure that the binding is as expected. You can block essentially 100% of the specific binding. And so really each step along the way ensures that the assay is being done properly and it's reproducible, which I think obviously in science you want reproducibility. In RO assay, it's something where it's very sensitive, and so getting that reproducibility can be tricky. And so to be able to demonstrate it shows all the work that went into the assay.
Yeah, and they definitely did, impressive amount of work in terms of looking at exactly how specific that PD-1 detection was in their paper, as well as looking at both healthy and disease state samples there.
Right. It seems like initially a lot of the work was done on samples that were not stimulated, just trying to identify what those baseline PD-1 levels are. Later on, some of the confirmatory testing was done on stimulated samples. Excuse me. And so let's see. I think going over to some of the items that you had as far as the agenda here, we've already touched on calculation, right? So we did that on a previous episode. We've got pre-analytical risks. We've talked about collection, shipment, transit, and so touched on staining, acquisition, data analysis, so on and so forth. And so here, really, it's just highlighting the ability to confirm the assay.
Yeah, definitely. This paper was really focused on that. I guess for the main portion of this podcast, we wanted to talk about it in the context of a clinical trial. What that calculation looks like. So basically needing to have enough blood for each sample that comes in for both generally a saturated and an unsaturated sample.
Mm-hmm.
And potentially, depending on the style of the RO assay, potentially two different detection markers in the same channel there, or in some cases, two different ones.
Yep. And I appreciate you jumping in there. If you couldn't tell, I had lost my place on my notes and was trying to follow up. Yes, thank you. You're talking about the samples. One of the things that was interesting, so we've talked about before RO samples primarily done in frozen, fresh matrix, fresh whole blood. The interesting thing here with this assay is they had done it on PBMCs that were then stored at -80, and they had demonstrated a stability of 153 days. Now, at that 153 days at -80, they had lost about 6% expression levels for key markers. I'm forgetting exactly which one they're basing it on, so apologies, it sounds so vague. But the point being is that they didn't lose a lot out at 153 days, which probably could beg to further extending depending on doing more time points.
That's actually incredible at -80. For some reason, I saw PBMCs. I just sort of assumed that it was going to be liquid nitrogen, but my goodness.
Well, yeah, I mean, most everything we do is at liquid nitrogen, just because it tends to keep samples more stable over time. I know in a previous role working with hybridomas, generally, if you had them at -80, by the time it was a year out, you really didn't have a hybridoma anymore. And so you always wanted to make sure to get those cells into liquid nitrogen. 153 days, I mean, not that quite, at a half a year, but closing in on it, right? And so to be able to show such a small drop over that period of time is impressive.
Right? Extremely impressive. And that does address some of the risks here that we mentioned with the pre-analytical. So definitely with RO, I would say in about 90% of cases we're dealing with whole blood. So then you've got everything from the time the blood comes out of the patient until it gets into the instruments themselves. So that is the collection process, the shipment process, anything that can happen in transit, and then once the tube actually gets there, how it's received. And if you have PBMCs, in theory, you could have that isolated at the site that draws it and eliminate a large amount of this variability. So if it's at all possible, then that is a great option to have. As you mentioned, this paper shows an example of the RO on PBMCs. I know we've got a couple projects ongoing where we're looking at that, and I've seen other publications in the literature. So I would love it if this becomes a more standard part of clinical trials.
Yeah, you and me both. We're seeing a little bit more of that recently. But certainly, if it were to become standard, you can demonstrate that the freezing process is not artificially altering your RO percentage. Then yeah, it's the way to go. So the other thing that they did was one of the other things, they did a lot of test scripts. One of them was on fixed stability, looking at formaldehyde treatment there, demonstrating that once you've got the sample stained, they're able to then sit for a while before being analyzed. This is certainly helpful if the machine goes down or that there's some other issue. You can still get good data if you can't put it on the machine right away.
Sure. And definitely with PBMCs, it does take a little bit of the risk out of the staining and preparation process as well, because then you can run in batches. But for a normal sort of whole blood RO assay, then the robustness of that staining process then becomes really critical. And making sure that everybody's fully trained on the assay before they start the preparation, and that all the appropriate procedures are in place, potentially reagents bridged and things like that as well.
Mm-hmm. Yeah, they did point out the variability. You mentioned somewhat on this as well for PD-1 expression, highlighting that there are a couple of non-assay related things it could be, whether it's the disease status, cell type. But the key here for us is certainly the method itself, right? And how robust you've created that method, whether or not it's been validated. And so when we talk about validation, it is about each one of these test scripts looking at within sample variability, within between sample variability, and then stability for samples here, if it were fresh, then or versus frozen. Fixed sample stability, where you're treating with formaldehyde, something along that line. And each one of these then demonstrates what sort of data, what you can rely on, and what you might expect to have higher variability from.
Sure. And, I like the fact that even though they are running in batches and that should significantly decrease the variability they're seeing, they're still using the quantum simply cellular beads. So very similar to MESF beads in that it provides some level of standardization, and then you can really compare easily from run to run. And that can be on the same cytometer or, in our case, even running in multiple facilities if there are regulatory issues or logistical issues with getting samples to a single site within the appropriate time frame.
Yeah, it gives you that understanding, that comfort, I guess I would say, more that you can interpret between sites, you can interpret if it's on a different machine. And certainly the field is moving more and more in that direction because it is important. Science is about having the appropriate controls, and so that's part of why that's there.
And the last thing, I guess, that I took away from this paper, and that is always an interesting part of clinical trials, is really sort of how to interpret the RO data once you get it. So, in some cases, the calculation is just per time point. And then some cases, you're looking over sort of a longitudinal time, what is the RO percentage as it goes through. And that should ideally follow the dosage regimen for the trial.
Right. Depending on what the treatment is, what receptor it's binding, how strongly it binds the receptor, whether there's receptor turnover. And so all of that information then gets taken into account to understand how the treatment is unfolding.
Right. And one of the things that I definitely noticed was just that, especially with table 2C, there were a number of analytes that were just below the limit of quantification. So just flagging that, that is one of the things that can happen. So as you had mentioned at the beginning, keeping the RO assays sort of as simple as possible is always a good idea. So if you're looking at larger cell populations like T and B cells, you can almost always get a reliable readout. But then with several of the cell populations, expecting lower readouts or more highly variable readout.
All right. Even in this one, they had pointed out that the readout for the NK cells, when they had done the disease samples, really wasn't robust enough, right? It just wasn't to get in those high enough numbers. But here, this is just one of those things when you do a validation, you do a qualification, even if you have a reportable that fails, that doesn't mean that the validation failed. It doesn't mean the qualification failed. It just tells you that that data is going to be a little bit more variable. It gives you an estimation of what that variability might look like versus other parameters that have passed, and you can be confident that they're within those windows of expectation.
Right. Yeah, definitely in previous lives, I've seen RO data actually go entirely negative, which doesn't entirely make sense. So always have to take some of these things with a grain of salt, especially when you're dealing with very rare cell populations there.
For sure. And we did talk about at one point looking at rare cell populations, blanking on the name of the paper. Would love to be able to mention. Perhaps what we could do is link it to the posting of this episode. But yeah, I mean, it is tricky when you do get down to those. That's why a lot of times for validations, what we'll do is set a minimum number of events that we need within the reportable to be able to evaluate it, right? And so if it is below that, then it's not reliable. And this can be dependent on essentially establishing an LLOQ and then utilizing that, or at times just in general, there may be a number that the client doesn't feel comfortable going beyond. And so even if we've got the LLOQ set to a certain number, they may not feel comfortable going quite as low, and so may set it a little bit higher.
Sure. It can make the data more robust and more trustworthy, but you may also lose a little bit of otherwise valid data there. So always difficult when, as you mentioned, clinical trials are quite expensive per time point, and want to make sure to maximize everything you get out of it.
Yeah, interestingly, I talk about that in the context to hear the number of events within a reportable. More often than not, if there is a validatable parameter that clients tend to be a little bit more conservative on, it is usually stability and the number of days that they have. So even if a sample had gone out to say 120 hours on a fresh sample, may choose to bring that back a little bit, and instead of 120 use 108, 96, whatever, just because there may have been a trend in the data showing that it's about to hit right at the point of being a validated parameter.
Okay. Excellent. I mean, that's most of what I wanted to cover about both clinical trials in general and RO assays on them. And this paper sort of in specific, I thought was an interesting exploration of that, right?
Absolutely.
Yeah. All right. So then, in terms of feedback from fans and questions, I did look at the YouTube videos, but did not find questions on the YouTube videos. I
Yeah, I
know we're
think,
getting close to that 20 episode minimum for Spotify, so hoping to get some more comments there.
is it 20 or 25? I was
Oh
thinking
no,
it's 25.
maybe 25. All right, we're still pretty far away. So, Adam, we may be doing another episode next week. We'll see.
We're
But
just going to have to start doubling up.
right? But definitely to all of our regular listeners, would encourage you to submit any and all questions you have, and we'd be happy to feature them on this section.
Absolutely. Did I hear you say that's like in goals or something like that? I kid. I don't want anybody thinking that I'm putting that in their goals.
Yeah, we'll have to see. I don't know. But
You may overrule me on that one.
we're going to have to submit at least 3 questions to the podcast. Yeah. No, but definitely look forward to interacting with fans and looking forward to getting some more guest speakers on in the near future as well. So once we have those names locked down, then we'll definitely feature people on the podcast here.
Sounds good.
Yeah. So in terms of other things, I know we just got done with Memorial Day weekend. Any big sort of outcomes there?
As far as outcomes, let's see. No, did some top golf. I think I only won one round out of 3, but hey, I don't have to win them all. But I did have fun. Interestingly, I'm not a golfer. I'll be the first to admit that. My father is the golfer in the family. But, I started out with a club that I usually use, and this is the Hybrid 3, right? And I was all over the place. I don't know what was wrong with me. I tried to drop down and use an iron because usually you're a little bit more stable with the irons, and it's okay. And then I decided, you know what? Forget it. I'm going all in. And I went with the big driver. And all of a sudden, my hits just straightened out and I was doing great. So I don't know. I guess I had to learn and go through the process. Honestly, I think I needed to step a step further away from the ball. And the driver forces you to do that because the club is a bit longer. Anyway, I digress. For anybody who is completely bored of golf, I enjoy doing that. That mini golf, pitch and putt. Occasionally do 18 holes, but for my stress level, usually I stick with smaller courses like pitch and putt. How about you, Brian?
Yeah. I mean, have you seen the movie Billy Madison? I'm seeing the need for a larger and larger driver here.
That's awesome. I could totally see you doing that. Now, have you hit the ball trying to swing like that?
Not quite yet, but definitely in the near future, I'll have to give it a shot. They did the spin move there, right?
Mm-hmm.
Yep.
Mm-hmm. And there's a new movie coming out. I didn't realize they needed to have a second Happy Gilmore, but I love the first one. So fingers crossed it measures up.
Yeah, indeed. In terms of the weekend, had my daughter's daycare graduation, which I didn't know was a thing, but is now a thing.
Congratulations.
Yeah, thank you. No, congratulations to me especially, because I no longer had to pay daycare fees at some point, right? So definitely a big celebration there.
This is absolutely what I was mentioning was congratulations to you for that.
I guess good job to my daughter for making it through daycare too. But yes, indeed, all the parents were very excited there.
That's really cool.
Yeah.
Any interesting foods this weekend?
A standard Indian fare. So always go for a vegetable korma at a local place. That's usually pretty good. So.
That sounds good. I usually go for chicken makhani. Get some cheese dosa in there. Oh man, definitely making me hungry now.
Yeah, luckily one of our colleagues is in from Lyon, France this week as well, so we'll be able to show him what Philadelphia cuisine is all about. So he's excited to try some cheesesteaks.
I'm looking forward to that. Can't go wrong there.
Right. All right. Yeah, so next time I know we're looking forward to some different things. One of them will likely be some CAR NK cells, but could also be some different types of CAR cells. So looking forward to our next episode.
All right. Yeah, that does sound like fun.